Okay, good morning again. This is Dharma Bennett with the State Water Resource Control Board, and this is the August CEDEN user group meeting. Thanks again for joining me. We just have two items on the agenda this morning, and the recording genomic sequencing metadata, and then a check-in on CEDEN 2.0 questions. And so we'll go ahead and get started with the first item. I'm happy that Tessa Foget, the QA officer for SWAMP, is here to help guide us through some proposed business rules that I added to the bottom of the agenda. So I'll hand it over to Tessa. Thank you.
Hi, good morning, everyone. Jeremy, do you just want to page down to show the instructions? Cool, thanks. So we are, I guess we are adopting this as kind of like a business rule for SWAMP, but we're just kind of sharing it with the entire group because it just might be helpful guidance for anyone else who might want to submit sequencing data to CEDEN. And I will say I am not a sequencing expert. My former staff, Charles Brook, wrote this up for us, but he has since moved on, and we're hoping to have another sequencing expert come on in the next couple months here for any real questions about this stuff. But basically, people who are collecting some sort of genomic sequencing data. So like we are starting in SWAMP to collect environmental DNA metabarcoding data. So basically you get this DNA from a water sample, and then you can associate it with different species based on the DNA. And the actual genome sequencing data is large. It's a big file, and we don't really have a great place for it in CEDEN, but there's already this federally existing database for sequence reads. So we're basically relying on that of where we're going to actually save the sequence data. So that's called the NCBI, National Center for Biotechnology and Information Sequence Read Archive. So the NCBI SRA. So that's already this well-established federal data repository for this type of data. So these instructions are basically, kind of tell you how you can upload your data to that, and then kind of how we would link it to data in CEDEN. So like we would still have, your field collection data, or if you had other related data in that project, like maybe you had actual taxonomy data, or you had just other water chemistry, or just field observations or what have you. That would also be in CEDEN, and then there would be kind of this metadata to link, so that if you found that project in CEDEN, you could then go look up the sequence data in NCBI, or vice versa. If someone was looking at sequence data in NCBI, then they could come to CEDEN and find the same, the linked project data in CEDEN. So that's basically what these instructions go through, is just kind of what to name things, so that if you're in one database, you can find the related data in the other, and vice versa. So I don't think we're really necessarily like making this some kind of rule for CEDEN, but it's more just guidance if someone is collecting some kind of sequence data, most likely environmental DNA at this point. So that, it's just kind of giving some guidance so people can find it and link it. So I don't know if there's any questions. Like I said, I'm not the expert, but I have gone through the links and kind of seen how it works. So I can try to answer. Marco, yeah, go ahead.
Yeah, hi, Tessa. I was wondering, like why is it that we're not saying like this is what everybody should do so that it is standardized across the state, whether it's swamp or CEDEN data. We just spent over a year working on the passive sampler guidance, and it seems like, we should be comparable whether it's swamp or CEDEN, and that way it is easier for people to find and associate data.
Okay, and maybe Jarma and I just hadn't confirmed that. Maybe that is the intention. Jarma, do you feel like should be like official business rule?
As you were describing it, it made sense just to have it as guidance. But I mean, I can see Marco's point, but we don't have any control over this other database, and I don't think it changes the data that are coming to CEDEN. It's sounds like you use them in conjunction to get a better understanding of what's going on, but the data that's held in CEDEN, we still have business rules for and things. So I think I'm at this point just thinking that it is guidance on how to, if you want to make the data more valuable or useful, comprehensive, to do it the way this is outlined. But
yeah, I guess I think what we were imagining is if people came to us with eDNA data or, whatever sequence data, and they were asking, can we put this in CEDEN? We would say, well, not the actual, you can't put the sequence data in CEDEN, but here's what you can do and how you can link it to anything in CEDEN. So, yeah, I guess it's we're not providing a space for sequence data. So it was sort of just like, well, here's how you could link it all together if you wanted to, but.
Yeah, I mean, I think it would be helpful to have it as official guidance on the CEDEN website, so like if somebody was to do this, that it's there and it's available, so that it would be easier for everyone. I mean, what you're showing, it says swamp/CEDEN anyway, so it makes sense to just make it official and make it available so that people can standardize if they come across it.
Yeah. And I think that was our intention. We would, after we discuss this with you all and see if there's any edits, then we would post it as guidance or on the CEDEN website.
Right, but we
so
have
I don't know how checking. I guess it's more semantics.
Yeah, if somebody asked me, do I have to do this? I would probably say, no, this is our guidance on how it could be stored and linked, but I'm not going to tell them that they have to. But I mean, for Swamp, Tessa, you guys are doing this method, right?
We are.
So funded.
So basically, if we're funding someone to collect sequence data, then we're going to require them to do this. So that's, I don't know, like for Swamp, we're calling it a requirement for anything that we're funding and paying for sequences. Then that's a requirement for us. But yeah, CEDEN's just a little different because they're not funding the studies. Okay, and then Tori.
Yeah, I'm sorry. I always do this where I don't see the second page. I apologize. So I didn't get to look at this before, but I think my first question is, if you're going into CEDEN, how can you tell if they have multiple data types? How can we tell that the project does have sequencing data? So you're saying it's linked, but from what I'm seeing on here, it's showing how to populate this other website so you know where it's coming from. I'm just wondering how you can tell when you're in CEDEN that it does have sequencing data in this database. Like, are we consistent with what we're putting in our own database in our own comments, or how are we doing that so people know that data's in here?
Yes.
I think I might be just missing I'm sorry if I'm missing the step.
No, and maybe that's not, while I thought we did have that written somewhere, but maybe it's actually not on this page. Yeah, because and maybe we need to add that. So, because what we're doing in Swamp is, I think it is in a comment, we're writing kind of like the code in the NCBI so that you could find it there. So Sorry, and I like skimmed this again this morning, but I hadn't, didn't think it through in depth this morning. So we'll make sure that's in there.
Okay, thank you.
Also in the same guidance, so it's all the
Is
whole.
there an
whole
actual SWAM/CEDEN template example for the genetic data? Not so much the sequencing, but like how to actually report the results that CEDEN can store?
We can make one. Yeah, because I know we worked on this because we were getting sequencing data in, so I know I have it. We obviously didn't get it all in this page. That's why we bring stuff to you all to point out the obvious. Okay, Adam.
Thanks. I think mine's sort of related to Tori's question. Is the intention that there will be like a dummy record essentially in CEDEN that then links via I think you said a comment field to like sort of points you to a record in the NCBI database, and then similarly in the NCBI there's a comment that sort of points people back towards CEDEN, because like you had mentioned, like, oh, if you have collected samples at the same time, then you might have like an existing project station code collection time. But if for instance you were only collecting sequencing data, are you putting some sort of record in CEDEN with no real result, but some sort of dummy record? I wasn't sure what the intention was.
Sure. Yeah, these are all great questions and why this group exists. So I think when we were coming up with this, we're doing an eDNA study right now, and that's going to give taxonomy information. So we're going to put taxonomic IDs in CEDEN, and then it would have something in I think in a note column or something, or comments or something that shows where to go find the sequence read. There's some kind of, ID or code or whatever to link up. So we can definitely provide that as a template or example. I mean, I totally not the DNA person. So that's, that project is kind of how this idea got started. It's possible you could have sequence data for something other than taxonomic IDs, I think. Maybe, or maybe not, but so it may not be like a perfect template for every possible project, but that's like kind of the most obvious, I think, of how people would be using it and wanting to put it in CEDEN for now. So we can have that as an example. Tori, was that another, or was that a from before?
Sorry, no, I didn't put my hand down. Sorry. I'm sorry, what does the output look like from this database? I've never actually messed with it. I'm just kind of wondering if you have an output, if we can see that in the example too, it'd be kind of nice so we don't have to create a login and everything. I'd be curious what it looks like and how they connect back and forth.
Okay, we can get that. I'm interviewing for my, backfilling this position next week, and I am so excited to get them on board, so I don't have
I
to
bet. I'm sorry.
be the person like, I don't actually know.
Okay, so it sounds like we have some edits to do, and maybe you could just find those data that have already been, you guys have worked on already, and I'll modify it. A couple different changes to be the CEDEN templates instead of the SWAMP templates, and we'll get an example of the output, so we can make sure that what's being suggested to link the two, to let people know to look in the other database is in there, but that we may not come back to this group until you have your new staff is on board, so we can work on it.
Yeah, so it might not be next month, but hopefully the month after.
Yeah. Okay. Great. If people have, since we will have probably some time, if you didn't get a chance to review the second page of the agenda and have additional comments or questions or edits, please let us know and we can try to incorporate those as well. And thank you again, Tessa, for coming to the group.
Thanks, everybody. Appreciate your comments.
And you're welcome to stay on for our CEDEN 2.0 discussion, which we will talk about next. Tessa is highly involved in that project as well because, as we have talked about, the new database will be a replacement for the SWAMP database as well. So definitely she will be impacted. But changing gears to CEDEN 2.0, hopefully that's opening. I haven't gotten to address too many other questions yet, but I appreciate them being logged here so I can work through them and we can all see the conversation here. So that's good. We talked last time. I kind of jumped right into this just to provide a little bit of background if you're not familiar, that CEDEN 2.0 is a project we've been working on for a couple years now to have to replace the current database. It will be also be replacing SWAMP so that the functions that SWAMP staff do now, the quality checks that they're doing now will be included in the functions of the database itself. And excuse me, I went into the system last time and I have shared the template that we currently have. That was one of the questions in this question log, and I provided the link to that. And we'll go there right now so we can just review that. So it is a, there's an on-screen entry, and you can also upload your data like you do now, and it runs through checks and flags what things do not that are not consistent with our format and our checking rules. So this is our format. Again, somebody asked what it is currently, and I provided this link to the posted format. It does still, I've highlighted these last columns. I guess that's brown, and we intend to get rid of those fields from the template so users don't have to provide those data, and that they will be picked out from the data that's already being submitted. So that's kind of why this is not being official yet, because we're still needing to make those changes there. So that was one of the questions. And I don't have all of these examples yet. I have a couple of them, but that I know we have in the examples that we've translated to the CEDEN 2.0 format. And so I will be, I guess, maybe just adding tabs to this to show those, or if there's a suggestion on how to show these different requests in different files or different tabs, or just a couple lines for each one, kind of like we have in our example template. Does anybody have a suggestion there?
Yeah, another worksheet would be fine, Jarma, and then you could have all of those examples in one worksheet or split them out, whatever makes it easier for you.
Yeah.
I think that personally the examples are just going to help me versus listing all my questions out. If I see it, it's just going to, I feel like it's just all going to come together versus having 20 questions for you. I think just having those examples will answer a lot of my questions.
If this was your entry, Tori Woodson, or whoever did the entry, if they're online, I'm not sure if you have reanalysis and re-exported diluted sample, what you put within the test type. Can you explain that scenario?
Yeah, so if you have a sample that is a reanalysis and it's a diluted sample, I'm just not sure. I would like to see the different situations for that test type field because it has, I don't know if it like concatenates those things together or how that looks.
Oh, I see. Yeah.
Because those
Okay.
two things can be for one sample. And I'm assuming they're like concatenated together or, put together, or I just seen one code. So I just don't know how that test type field looks.
Okay, I see now, because they're two different things.
Yeah, because there's multiple things within that test type field from what I from the information that I have, or which is minimal, but I just feel like it was capturing a lot of information there.
Okay, yeah. That one we have to, I don't know that I have any of those examples in our current format to translate, but we can work something up.
Tori, would you have a specific data example, like from a template?
Well, like for example, our CEDEN template doesn't have reanalysis information. Like we don't store that. So that's like a new information that we normally deal with it by not putting in the we decide whether we want the original analysis or the reanalysis, right? But we've never called out that this is a reanalyzed sample versus like showing in the comments, talking about it in the comments. So I feel like there's no comparable coding in the current CEDEN template that indicates this is a reanalyzed sample. They chose to utilize the reanalyzed sample versus the original, other than we usually put that in our comments.
We do have, Tori, this is Stacy, we do have QA codes. Those are the PJN, PJM codes.
Okay, yeah, we don't use those.
Yeah.
I was just thinking, like, to help JARMA, maybe if you just took like an example data set of a few records and then just make up like a dummy reanalysis, and so you can just like highlight like this is the initial analysis record and this is the reanalysis record, both in the CEDEN template format, and then JARMA could use that example to show like how it would be reported in CEDEN 2.0.
Yeah, and I think we decided for CEDEN 2.0, they don't want to analysis like we've always done in terms of, as a program, we never want to have the original and the reanalysis reported. We always choose between the two and make a decision at that point before putting it in the database. So in terms of seeing them together within a template, we always remove one. Does that make sense? We personally never put both in the database, and I thought that was what CEDEN was trying to do as well. But I feel like this test type field is calling out that it's a reanalysis, like what Stacy was saying. It's probably that PJM QA code.
Okay.
Does that make sense, Marco? Like I would never put them together, but I can put examples together. We get it from the lab all the time.
I guess I'm just saying if CEDEN 2.0 is going to allow it, and there's going to be an example data, that I thought it might be easier if you just helped JARMA out and gave her an example data set that they could then use to show how it would be reported in CEDEN 2.0.
Yeah, I've got lots of it.
Okay, great.
I can give you multiple data sets with all of these things in it.
Perfect. Lisa?
Just a I don't know, comment, I guess. Some labs and most of the ones that I've ever worked with don't typically report out 2 results for one sample. We make that decision based on the quality control criteria that we have to follow by the method to determine which result we send you. So unless we're asked to provide all results, we combine them prior to going into the CEDEN that I send out to somebody to upload.
Which I think is very good.
Yeah. Okay.
Yeah, and that's the
Yeah, very good.
intention.
Yes.
That this is Tessa. So we don't want multiple results for a single analysis. So it's more just that you wouldn't give the initial and the reanalysis. If you needed to do a reanalysis, you would then just report that one result, that final, as the reanalysis. It's more just
Well,
letting us
sometimes you
know.
can
Or-
have
yeah.
like when you have multiple analytes in a test code, you can have like we report the lowest valid result. But if you have a dilution, we'll have a reanalysis for the dilution for 1 or 2 compounds. And we merge those. You have the dilution factor in there, but you'll just get one result. Does that make sense?
I think so. Are you
Like
you're
you have an
saying
8260 and
yeah,
most of the analytes will have a dilution factor of 1, but then you might have a couple like benzene or toluene if you have like TPH contamination that have like a different dilution factor, but you won't have 2 results for benzene. You'll only have the 1 result for benzene in
yes,
there.
yes,
And we just call it the same. Well, some labs call it the same lab number. Some labs will designate it as a second lab number with a like some kind of a like an RE or an A or a B, just to show you that there were 2 runs.
right, because you had to dilute for some to get some of those.
Yeah, but when it goes into the CEDEN template, it all goes in just as the one single sample.
Right, but don't you give the dilution factor for the analytes
We
that were
do.
diluted?
We do.
Okay, so
For the
that's kind of,
analytes.
this is like where you would give, well, it's not where you would give the dilution factor, but this is where you would say if it was diluted or not. So I see what you're saying, like,
Yeah, it's just
would you call that a reanalysis? It's like you knew
yeah.
you had to do that from the start, but you knew you had to have 2 runs.
Yes. Yes. So
Okay, I
do you want
see
us
the
to report that as
question.
2 different
I
lines?
don't have
One
the
is
answer
a
yet.
yeah, okay.
I don't think you'd ever want 2 different lines. You just want one result.
Yeah,
Yeah,
we
I think
only ever
Whatever you're putting in that test type to indicate whether it's a real analysis or not. But I think as CEDEN as a whole, you always just want one because otherwise
definitely.
no one knows what the value, the lab is making a decision on what is the best choice, or the QA officer is making a decision on that. So you should go off of that.
But then
Yes.
do you just
So I'd
call it
never have to.
diluted, or do you call it diluted reanalysis?
Right. What do you call it?
Yeah.
So we'll come up with some guidance on that now that we understand the question.
Okay, Adam lowered his hand. Okay, run. Are you telling me? Okay. Adam, your hand's up.
Yeah, I was just trying to say, like, I think everyone's on the same page that it's just one record. And I feel like the way you've written this up it's complicating it a little bit more than it needs to be, in that it's just like if a single result is both a dilution and a reanal, like it doesn't matter about the multiple analytes, same method, different dilutions. Like it's just like if any one record, multiple test types as we currently think of them can apply to a single record, are we going to have, I think what Tori was originally asking about is like, will there be a series of concatenations as far as valid? Is it going to turn into something more like prep preservation lookup, where you have just a whole bunch of different combinations? Or is there a different way that we're addressing it?
Yeah, so on dilution underscore reanalysis, that's was supposed to be the concatenated option.
Yeah, for sure. I just meant like the multiple analytes, same method, different dilution, like that. That I think is kind of the side that it's more about a single result with both a reanalysis and a dilution.
Yeah, I think it was more like the lab may not even like the example she described is like the lab wouldn't necessarily consider that a reanalysis because it was like they knew from the start they had to do that dilution. So I think maybe giving some guidance on like what is considered a reanalysis versus just like needing to run different dilutions because different analytes come in at different levels. Was my understanding.
That wasn't how I read it, but okay.
Well, I'm not, that was my understanding of the description, but was that Lisa, or if you could confirm if that was the question?
Sometimes we know, sometimes we don't know whether we'll need to do a dilution on a sample. So I think part of this is nomenclature. Anytime we do a rerun, we call it a reanalysis in the lab. Not necessarily just another run for a dilution. But if you're talking reanalysis because it fails quality control or something like that, and it's out of hold time, that's about the only time we would ever make a notation that it was a reanalysis for our clients. Just because the reanalysis part is part of the the procedure. It's built in to the method.
So it sounds like we need a little more definition on what we mean by reanalysis and what
Yeah.
is
Yeah.
considered reanalysis or not.
Yep. Yep. Because it could be, I mean, you guys might want to, there's other tests that I don't know about, like the taxonomy that might require a reanalysis. And it might actually say, this is a reanalysis in the method. I don't know because I'm not familiar with that.
So we're just worrying about chemistry for now.
Yeah.
We'll cross that bridge when we get there.
Okay, so hopefully I reworded it to more discuss that we need to give more definition to reanalysis with a specific example of is it considered a reanalysis to rerun it for dilution? And we can work on that. Adam, is your hand up again?
Yeah, because I think the original question still is there, which is, I think the examples we've been given so far for test type, there was one for dilution, one for reanalysis, and the question is, will we continue to populate the lookup list to handle combinations of what look like currently like single values for test type?
Yeah, that was my original question, Adam. So you've said that a little better. So thank you. Give a good example. Thank you.
And yeah, we can confirm that list. I don't have it offhand, and confirm we have the bases covered that we think we need. Corey?
I forgot to, do you have another update also on timing? Can we also add that too? Just timing of when you guys, I know it's getting closer to fall. I'm just wondering if you have more of a timing estimate, if you guys are getting close or when this will be rolling out.
Yeah, we like
I know we say fall, but
yeah, we, like I said, we need to get those the fields that I noted removed for our official template, and until we have that version that's tested and good, then I don't have it's not official yet. So and then we'll get an implementation period before it's required, like we have been doing when we make changes to the template.
you'll probably know a couple months before, like a month before. It's not like gonna happen next month or anything, because when I think of fall, I think of October, but
It's not going to happen next month.
okay.
We'll
Just want to make
definitely
sure,
give
because
fair warning.
timing is a
Right.
very
I
big
mean,
thing for our clients, and my boss keeps asking me, and I'm,
right. I mean, and as I said, we'll have the 6-month implementation period. So even if we come out with the official version October 31st, so it's still October, then they'll have 6 months from then to,
budgets
adjust.
and everything go for like years, so it's hard to 6 months. Okay, thank you.
Let's see. Lisa? Oh,
This is just no, sorry, clicked the wrong thing. This is a follow-up on the last question. Is Firstsoft going to be doing a training for, because this is an Equus database, are they going to be rolling out a training on it or having something on their website similar to what they've got for like the clip that they did?
They are working on a series of training videos for us. We requested multiple shorter videos instead of one longer session, so that, in hopes that you just go and review what you need and then move on to the next part and review that. And we'll be posting them on our website and having links to them within the application.
Okay, and you'll have all the links to the valid values and all of those things, the lookup lists.
Yeah, right now they're within the application, and that's where they are, dynamically updated, and yeah, and there are buttons to go to those widgets to view existing vocabulary.
Okay, all right, thank you.
Cassandra?
Yeah, I was just going to follow up on the earlier row about creating templates with data in them. I guess I was hoping, if it's possible, if you're going to create templates with example data, if you start from a CEDEN template, it would be nice to see both, so you can see what the data looks like in a regular CEDEN template and then see how it's been applied in a 2.0 template. Like I could see that as valuable information. So if you're trying to hone in on a field, see where it went from one template to the other.
Yeah.
And then my other question was about row 13 on the user interface. I know we still use the AQT a lot to filter in on a project. And if we're trying to see the chemistry data, maybe that's still going to be on the AQT. How is that going to mesh with the 2.0? How would we be able to do that? Or have you guys thought that through yet?
We have thought as far as knowing that we need to provide an access point where you're accessing CEDEN 2.0 data and old current CEDEN together, so you can see through time what the data are saying. We haven't started what that access looks like. We have worked on a tool that converts current format to new format. So I envision that we can kind of leverage that work that's already been done to kind of transform data to do the same thing for the backend data, whether and make that accessible. We'll probably start with just a flat file of data that you can access together, and then work towards more of a dashboard interface where you can pull those data together.
Okay.
If many years ago, probably 7 years ago now, maybe, like we had, before we started a new CEDEN project, we were going to start with a new query tool project because I've gotten many complaints about the current query tool, but then we thought, well, if we make we start with the data out, and then we change the input, the data in portion and the data storage portion, and what will that do? That's probably not a good sequence. So we put that on hold and are now longer than anticipated, but are working on the database. So it's always been a thought that we would redo a query tool to be more what people needed, and it will take some iteration steps. So like I said, we'll probably just start with tabular data together and then move to something more interactive, like people are used to seeing nowadays.
Okay. Do you think that that's going to happen though before CEDEN is requiring data to go to 2.0 so that we don't lose any functionality?
No, I think we can. We'll have one by the time we have the other. Our plan is to start using before it becomes required for general data providers that will be using it internally. So we'll have some data to work with that is in the system at that time, and can be working on those tools during the implementation period.
Okay, thanks.
Marco?
Yeah, hi, Dharma. The RDCs requested a while ago the ability to continue with the weekly transfers of chemistry data along with the habitat and taxonomy and everything else. I was just wondering what the status is of that.
I have not heard back from management on that. So I don't have a status, sorry. Okay, so some of the other, there was a request to see the EQAP format, and it's not complete as noted it kind of in the request, but I think we can provide. So I'm showing it now, and I'm looking for which field I have I don't remember now. The filter on, but to give you an idea of what it looks like, there are several tables. So the one that we're on now is the control limits table, and everything is in the same QAP that we have. And there's going to be one QAP at rollout, and it's the this CEDEN ambient surface water master, and this will provide the basis as we move forward. We can work on putting allowing data providers to provide their QAP and to get it translated into this workbook format that can then be used against their own projects. But again, that'll take additional steps. So we're all going to start with this one first. And you define the matrix code and the method. And this constituent index is our constituent. And then it has a bunch of columns. Again, I think we'll post this so you can look at it in more depth. But and then it allows you to specify the limits that the bounds in which we want to see the results within. Otherwise, the data will be flagged. Tori?
When you say you're going to start with this, but when we start submitting our data, we will have our values in here.
No. You'll it'll start by being checked against this this QAP.
So when will we be allowed to go against our own QAPP?
I don't have an estimate on when that process will be in place, both in terms of, because it will be a manual process to translate it into these this table format, and then the step that allows a provider within their upload to specify which QAP they want. We need that in place as well, and we don't have that piece in place yet.
So even if we filled this out and gave it to you, it still wouldn't work because you have something has to indicate what you're evaluating against.
Yeah, which set of tables you're looking to.
Okay.
Adam.
Sort of following up on Tori's question, is there, like, are you anticipating having that capability by the time that this is launched, like that's sort of at the end of that 6 months period?
It will be after that.
Okay. And then the other question I had is, we have something like this already set up internally, and we have certain things as sort of optionally filled because, especially around like analytes, because some QAPs will have, or some methods, I guess, will have MQOs for the whole set of analytes, and some have analyte specific MQOs, like the DoD, like PFAS requirements. And I wasn't sure, like I think I saw that the analyte name column was empty, and I wasn't sure if that was optionally empty or how it was implemented in the system. Or chemical name.
Oh, I can.
Tessa, do you want to? Thank you.
Sure. Yeah, so we can leave it empty, and then it applies those limits to any analyte with this method and matrix. And then we can also add additional rows with specific analyte names in there. That would, so then you can have different limits for those specific analytes. So there's a few where, like we've got some of the nutrients pulled out of like the other anions because they have so the limits are based on swamp MQOs, and we have different limits for nutrients versus just like the other anions. So those are like what John was showing is those are specified for these nutrients that you can do by 300.0. So yes, we can have specific ones and then a general one that applies to anything else that's not specified for that.
Okay, great. And then
Yeah.
the other thing I was going to ask is like, I could be wrong, but I'm assuming the current swamp clap doesn't necessarily have MQOs for every method in CEDEN.
Totally.
But
You are right.
but the EQAP will need to have something for every method. So is it just assuming like a default set of MQOs that it's going to start flagging data on, even for, you know, for methods that Swamp doesn't currently cover?
Yeah, that is what we're doing so far. And, you know, we're just hoping to continue adding to it and specifying stuff where, you know, like especially like where methods specify things like for PFAS or something, but like in general, like RPDs are all set at 25%. And then if it's something we don't have MQO specified for, percent recovery is generally being set at 50 to 150.
Okay. And so is there any ETA at all about when we'll be adding sort of new QAPs?
No, but we'll try to get you one.
Okay.
Because we want to know that too.
Okay. Because then the other thing, like, do you guys have a handle on sort of what the harshest flags that are being applied for being outside of MQOs? Is it the equivalent of something like an EST now, or can you actually get a straight reject auto flag based off of
nothing.
Okay.
We won't reject data.
Okay.
So yeah, all data will still be in there, and it's just an addition. It's more like a QA code, I guess, but it's a separate column from what the QA codes the lab provides.
Okay.
For not being within the numeric value specified.
Right. So it's like,
Yes.
you know, if we're internally evaluating data to an actual QAP and have, you know, for whatever reason it's rejected, that'll go in QA code. The auto flagging will have a separate column where potentially it gets an EST or whatever, but the fact that it was originally rejected or lab rejected, you know, QA officer or lab rejected is still maintained. Okay. And then it's
Yes.
never auto rejected. Okay. Thank you.
Correct. Yeah.
Thank you.
Okay, Lisa?
I just wanted to ask if the new CEDEN and the new QAP is going to address the fact that some analytes don't really have an MDL.
We've been talking about that. So like it's in this spreadsheet Jarma showing it's possible that we can have you could put in a check for MDL, but we're basically bypassing that check because we know it can change for dilutions and other things, and it's just not something we feel we need to evaluate. But we are also trying to figure out what to put in those fields for methods. Like when you're reporting the data, we're trying to figure out what should you input in those fields for parameters that don't actually have one, like some of the solids analysis or other things.
Yeah.
Or like alkalinity or no, I forget whatever ones I'm thinking
No, there are
okay.
a lot.
My brain is working.
Yes. Calculations.
We know
Yes.
that problem exists, and they're
Yes, so that is something we're kind of talking through right now and to provide guidance on.
Okay, thank you.
okay. How about we do one last question from Tori?
In terms of priorities, for what Equus is working on to fix, is this more of a priority than, say, your next data type, which is field?
I think our working after we get this actually in use, our next priority is field above the
Above the EQAP?
Mhm.
And then is this a priority or is it the next data type?
I think I think we're trying to do parallel efforts. And so I think we'd be able to get the EQAP, the multiple EQAPs in before we do the data type after field.
Okay, thank you.
Okay. So we're just about out of time. Thanks for all your questions and making sure we're thinking these things through. We have a number of action items to follow up on those questions that we've been logging and providing data. So if you, again, Tori, if you do have examples that you want to see how they look like in what they look like in CEDEN 2.0 format, that would, is
Yeah.
half my battle. So then I don't have to go
Yeah, no,
looking
I
through.
can, I'll have one of my staff compile it next week and we'll get it to you by Friday with all those different examples and it should be fine. I'll put in the CEDEN template. We normally, a lot of our stuff is in swamp format, but
Oh, small
will-
format is fine. Don't spend your time
well,
changing it
if
to
you want to
CEDEN.
show the group or display with the group, it'd be okay. It won't take us much time. I think I've got a
Okay.
transformer for it
Okay.
too. So
Okay, then that's great.
yeah, just to make it easier for you. Okay. Okay. Thank you though, Drummer. I appreciate that. My visual learning will help with. I just need something to look at versus words.
Yeah, understandable. Okay, well, we'll wrap up and thanks for joining us again. And we'll talk to you in September. Take care.
Thank you, Jarma.
Thanks.
Bye.
Thank you, Trevor.
Thank you.
Fantastic.